propagation in case of grapes. Grape is propagated mainly by means of cuttings. So, it is propagated mainly by means of cuttings. We have discussed that it is a vine crop that is climber in nature, and this is the primary branch, which is also known as arm. Then this current season shoots. Current season shoots are known as can. Current season shoots, as you know, in case of grapes, the shoots and inflorescence, vegetative shoots and inflorescence develop simultaneously. And this 1 year old current season shoots is known as can, and these cans are pruned during the dormant periods. And in case of, in case of northern Indian variety, the cans are pruned for one time, that is in the month of January. Then in the South India, the cans are pruned in the month of October and April. That's why, this in case of southern India, the flowers, the plants bears fruits for two times. That is known as October flowering and April flowering. So, April flowering means by March you have to prune. Here by September you have to prune. Then next month there is a flowering. Flowering means the vegetative shoots and flowers appear simultaneously from the nodes. This is the node, say, this is the branch, these are nodes, these are the leaves. Then here the inflorescence. Inflorescence will develop. So these cans are cut into pieces about 15 to 20 centimeter length. That means these are the branches, 1 year old shoots, technically known as can, cut into pieces about 15 to 20 centimeter and make in bundle of 50 or 100 piece, 100 numbers. Then they are these cuttings 15 to 20 centimeter length having 2 to 3 nodes, make into bundle of 50 or 100 numbers, then kept in peat for about 20 to 25 days. And covered with soil. The cuttings are placed in the soil pit for about 20 to 25 days for better callus formation. What is callus? Callus is the mass of parenchymatous cells. Callus is the mass of parenchymatous cells developed at the wound tissues, base of the cuttings, having the capacity for formation of root primordia. That's why there will be development of the roots. So callus is the mass of parenchymatous cells developed at the base of the cuttings, having the capacity for formation of root primordia. Sometimes for better root formation, the cuttings are treated with 1000 ppm or 2000 ppm indole butyric acids, IBA solution, indole butyric acids, and in some cases it may go up to 500 ppm. So, the dipping time in the IBA solutions, indole butyric acids, indole butyric acid is not dissolved in water. While you are preparing ppm parts per million, while you are preparing this IBA solution, first take the measure, say 500 ppm means 500 milligram of IBA, it is a powder, dissolves in about 15 to 20 ml of ethanol. It is dissolved only ethanol, ethyl alcohol. Then make the volume up to 1 liter. 500 milligram dissolves in 1 liter. Already we have taken the 20 ml ethanol. So, it is completely dissolved. Then make into volume 1 liter. So, there will be 500 ppm. Here in 1 gram or 1000 milligram, 2 gram. But when the concentration is more, then the time of dipping of the cuttings in the solution is less. Say here it is 20 to 20 second. Here it is 10 to 12 second, and it is 10 to 12 second, then 20 to 25 second, then it is 30 to 40 second. When concentration is less, the time of dipping is increases. Then these cuttings are treated with IBA solutions. Then you will find there is a, say this is the cuttings, this is the one nodes, another nodes and another nodes. Then you will find that roots are proliferated in good numbers. Sometimes while kept in the soil paste, before that we are dipped the cuttings. Then you will, while removing, then you will find there is a well developed roots, and when there is a root initiation takes place, with proper care you have to place in the nursery. Then 1 year old shoots, 1 year old cuttings, because the cuttings are placed in the nursery. Now 1 year old cuttings present in a nursery are suitable for planting in the orchards, and in case of grapes the orchard is known as vineyards. So it is known as vineyards. So at a spacing of 3 into 3 meter, 2.4 into 2.4 meter, then 1.8 into 1.8 meter, or 1.8 into 1.8 meter. That is in case of hedge system. That is in case of hedge system. Then in case of Nepean system, then it is telephone system, and it is for Bauer system. So these are training system of grapes. These are training system of grapes. Yes. This is Bauer system, BOWER. Bauer system, then telephone system, Nepean system, KNIFFIN, then in hedge system. So, in case of grapes, training and pruning is must. And after planting, within 2 to 3 years, within 2 to 3 years, the plant is trained to a particular system, depending upon the variety. The plant is trained to a particular system depending upon the varieties within the 2 to 3 years. What is training them? Training is a pruning operation. What is training? Training is a pruning operations performed in the food crops. Performed in the food crops to give the future shape of the plant or to give the future framework of the plant, to make the future shape of the plant. Then what is pruning? Then pruning is an art and science. Science is there, but it is an art also. Pruning is an art and science of removal of or cutting away or removal of unnecessary plant parts. Removal of unnecessary plant parts. What are the unnecessary plant parts? Dry branches, damaged twigs, excessive annual growth develops during autumn season, rainy season and spring season, excessive annual shoots. Then unproductive branches and the branch which is heavily infested with disease and pest. And the branch which is heavily infested with disease and pest beyond under control. When there is an attack of pest and disease, you have to go for control measures. Select the suitable, diagnose the pest or disease, select the suitable pesticides or fungicides, then spray it. When the particular portion is beyond control, then it better to remove it. That is also one type of pruning. So we have to train in different systems. So in grapes, there are 4 training systems. Head system, number 2 the Kniffin system, then telephone system and Bauer system. For head system, a small head is prepared with 3 or 4 branches at the top portion. That's why it is head system. The Kniffin system, 2 pair of shoots are allowed to grow at a particular length. That is 60 centimeter height and 90 centimeter height. This is the base portion, ground portion. Then telephone system, similar case, up to 90 centimeters, then 1 pair of opposite directions developed. Then these are removed, again another 1 pair in opposite directions. Then the shoots are allowed from these branches, just like a telephone wires. And these are also supported by wires at a particular distance. Then in case of Bauer system, in your practical manual, all these diagrams are there. Bauer system up to 90 centimeters, allowed 2 branches develop in opposite directions. Then another 3 pairs in opposite directions, 3 pairs. Then these are supported by some crisscross net wires fitted on the bricks pole or iron pole. It is above the heads, that's why it is known as Bauer system or overhead system. Bauer system is also known as overhead system. That's why depending upon the variety, again in case of vigorously growing variety like Anabi-Shahi, Bangalore Blue, Pearl It, then you have to go for Bauer system, planted at a 3 into 3 meter system. Of course, if the in Kniffin system, if the Pearl variety is grown, then the spacing is a bit wider, 2.4 into 2.4 meter. Then you have to go for weeding as and when necessary. Then irrigation. The vineyard should be irrigated during dry periods. During dry periods and after harvesting. And after harvesting at an interval of 15 days. And during fruit development periods, because especially in northern India, the fruits are grown under irrigated conditions, not as a rain-fed conditions. So, only at the dormant periods, when we have pruned the plant, generally December, January, no irrigation is allowed. Then from the first week of February, when there is an increase of temperature, sprouting starts in the can, then irrigate the field at an interval of 15 days. And after harvesting, irrigation is necessary because you have to rejuvenate the plants. Means by, say, if you consider the North Indian conditions, January you have pruned the plant, February there is a sprouting of shoots and inflorescence develop. Then fruit setting takes place, flowering, fruit setting, fruit development. It is continued up to middle of May. Then from middle of May, fruit is harvested till end of June. Then the plant exhausted. Then you have to go for first weeding. Then irrigation. Then manuring. Then plant protection measures. Why it is essential? In case of mango, in case of guava, in case of lychee, after harvesting, we are least concerned about this growth. Only at the autumn season, then we are perform the other operations. But here you see, in case of grapes, the flower bud differentiation takes place 6 to 7 months before its opening. Flowering is in the month of February, so by August, September there is a flower bud differentiation. That's why after harvesting you have to take proper care of the plant, then there will be more flower bud differentiation, means there will be more flower. And at the time of, again care should be taken regarding the pruning stage. Means, say, this is the can, right? This is the can. If it is a parlet variety, say delight variety, the farmers have to know the variety properly and he should have the good know-how about the variety, because in case of parlet, delight, cardinal, like that, the flower bud differentiation takes place. This is the original shoots, right? Then from here the new shoots can is developed. So it is the flower bud differentiation takes place in third or fourth bud, fourth node. But if it is Thompson seedless variety, then it is takes place in seventh and eighth nodes. So at the time of pruning, after, if the variety is parlet or delight, then after keeping the fourth nodes, then fifth nodes above, that should be detached. And in case of Thompson seedless, after keeping 8 nodes, then above portion should be removed. Otherwise, if you cut at the lower stage, 2, 3, 4 bud stage, then the flowering will take place in the nursery. That's why the flower bud differentiation condition depending upon the variety, and takes place 6 to 7 months before its appearance on the shoot. Hence, after harvesting, the plant should be rejuvenated properly, take more care for proper flower bud defenses. Then you have to go for manuring. In case of young plants under northern Indian condition, this amount is that 15 to 20 kg FYM, (500 gram) ammonium sulfate, ammonium sulfate, then 750 gram SSP, then 600 gram MOP. But in case of bearing plants, 30 to 40 kg FYM, 800 gram ammonium sulfate, then 1000 gram SSP, then 800 gram MOP, muriate of potash. And if it is the can, northern India, can is used, then it is 1200 gram. But in case of South Indian conditions, calcium ammonium nitrate, can, in South Indian conditions, for 3 to 5 years plant, 20 ton FYM, 500 kg nitrogen, 125 kg phosphorus, and 500 kg potash per hectare per year. But in case of bearing plants, this is 20 to 30 tons per hectare FYM, then 500 kg and 500 kg nitrogen, phosphorus and potash. The manures are applied in 2 split doses. Once after pruning. First application is after pruning. It is as a basal dressing, not in ring methods. That is below the canopy area. Give a light hoe, give a light hoe, that is make the soil loose, and apply the fertilizers after mixing and covered with FYM. The next is after harvesting. Then plant protection measures. Plant protection measures, the important pests are flea beetle, thrips, sapphire beetle, termite, then wasp and honeybee. Wasps and honeybee. So, flea beetle and sapphire beetle, they are generally feed the leaf, young leaf, and thieves, they are sucking insects, suck the cells from the young shoots and leaf. So, they can be controlled by spraying 0.1% dimethoids or Rogor or 0.15% dimecron. Then regarding the termites, it damages the base portions. So, it can be controlled by application of 5% malathion powder. That is, if there is no termite attack, then as a prophylactic measure, apply 20 to 30 gram per plant. And if there is attack, then you have to go for 50 gram per plant. Applied at the base, that is, just give dusting, applied by dusting procedure. Then a wasp and honeybee, when there is attack of wasp and honeybee, need not spray any pesticides, because this is also one type of maturity index. Means when the berries are soft, color break, that is known as horizon stage, color break, soft, swollen, and this becomes sweet and tasty. Then the honeybees fall on the buds and sucks the juice. So, this is the indications when this wasp and honeybee visit the vineyards, means the buds are at the ripening stage, better to harvest. So, this generally the farmers, most of the farmers, they are determine the ripening when there is a visit the honeybee and wasp. Because no pesticide is allowed, because they are generally beneficial insect, and if you even, but if you are not properly harvested after attack of the honeybee, then there is a vast damage. As you know, once the berry is punched or cracked, then the berry pulp is a good media for growth of the microorganisms. So, within 10 hours, 10-12 hours, there is a growth of the fungus, and whole the bunch is damaged, because it is, these berries are so compact. Once one berry is damaged, then within 1 day, all these inner berries are damaged due to fungus attack. Then diseases are powdery mildew, downy mildew, and most diseases, serious diseases, anthracnose. Powdery mildew, that is, these all are fungal disease. This can be controlled by spraying of 0.2% Bevestin. Downy mildew, it can be controlled by spraying of 0.1 to 2% Blitox and or keratin. And anthracnose, it is a serious disease, not only damage the leaves, shoots, and also berries. So, and mostly in the neglected orchards, improper managements, and more humid conditions. Then that can be controlled by spraying of 0.2% Blytox. Then the techniques for quality increase. That is quality of the berry. First is crop regulation, mostly by pruning. Crop regulation, then IBS, that is spraying of growth regulators, thinning of flower, flower thinning. Of course, the flowers are so small and they are compact because in one bunch 50 to 100 numbers average berries are there, and individual berry representing one flower. So they are so compact that at the time of flowering it is not possible. So only after berry formation, that the fruit sets, with the help of a scissor, you remove the inner undeveloped berries, that is thinning of flower. Then girdling. And another one is use of PGR, that is plant growth regulators. So crop regulations mostly by pruning in a proper trained plants. Thinning of flowers, that is just to increase the size of the berry and weight of the bunch with the help of a scissor after fruit seeds. Generally the inner branch, inner berries which are compact and they are generally underdeveloped. And girdling, this is also one practice we have discussed in the mango, no? That a ring of bark is removed at a selected shoots, selected can. To increase the rim of bark means the phloem tissue. Then the carbohydrates which is transported through the phloem, that is restricted, and above the girdling portions there is a full of, there is, this carbohydrate concentration is increased. As a result, there is more flowering, cause more yield. Then use of PGR. Use of PGR, that is 30 to 40 ppm gibberellic acids, GA3, gibberellic acids, that take in a beaker at fruit development stage, generally after 1 month of fruit set. Take a 1/3 of the, take a beaker and contains 30 to 40 ppm of gibberellic acids. Then it dips the bunches for about 10-15 seconds. Then jerk it, and in this way you can dip the number of bunches. So the farmers, grape growers, generally they are giving the containers to the children, just in the, especially in this Nippon system, Bauer system and telephone system, because they are hanging. So just simply dip it for 10, 15, 12 seconds, then jerk it, then again the same, soak the other bunches till the empty condition of the containers. Then again take another solution, and in this way you have to apply. Here no spraying, only you have to dip the young bunches after 1 month after fruit seeds. Then this physiological disorder, that is, one is saltberry, and another is flower or fruit drop. Saltberry, that is in a bunch, this is a peduncle, carp stem, rachis and berries. These are the berry, right? there is a proper development of the berries, but some berries will be small. Some berries will be small. So, short berry means all the berries of the buds are not properly developed. All the berries of the buds are not properly developed. Some berries remain small, green and hard. Remain small size, green and hard in nature. And that is mainly due to boron deficiency. Boron deficiency. And so here we have to go for 0.2% borax spray or 20 to 30 gram borax per plant. 20 gram, we write 20 gram. 20 gram borax per plant as soil application. This is all about the grape. So grape and grapefruits, they are two different fruits. What is grape? Vitis vinifera, belongs to the family Vitaceae. What is grapefruit? Citrus paradisae, come under Rutaceae family. This is a citrus crops. Grapes, it is a climbers. Grapefruits, it is a tree type plant. Grapes come under berry, it is a berry type of fruits. Grapefruits come under Hesperidium. So in this way you have to differentiate. So now then another fruit we will discuss. Jackfruit. Scientific name Artocarpus heterophyllus. Belongs to the family Moraceae. Type of fruits Solosis, come under multiple fruits. Edible portion of jackfruits bract, perianth and seed. Bract, perianth and seed. Jackfruits, it is a tropical crop, perennial in nature. Tropical crop, perennial in nature, evergreen plant, good spreading behavior, evergreen plant, good spreading behavior, and deep root system, extensive and deep root system. Good spreading behavior and deep and extensive root system. Long leaf plant, survive 50, 60, 70 years. Jackfruit is indigenous to India. Indigenous to India. It was also originated in Malaysia, Burma and Brazil. Besides the Artocarpus heterophyllus species, some other species are also available. In these areas, they are Artocarpus sempeden, DEN, Artocarpus altilis, Artocarpus lacosa, Artocarpus sempeden, DEN, Artocarpus altilis, Artocarpus lacosa, Artocarpus hirsuta, Artocarpus rigidis. So, Artocarpus altilis, it is known as breadfruit. Artocarpus lacosa, it is known as monkey jack. It is a evergreen plant, produce chloriflorous flowering nature. Chloriflorous flowering nature means the inflorescence is developed directly from trunk and main branches, that is from primary branches. In other food crops, the flowers are borne either terminal ends or axillary buds. So, if it is terminal ends, then that bearing habit is known as terminal bearer. If it is developed from the axil of the leaf, then it is known as axillary bearer or lateral bearer. So, here these flowers appears directly from the trunk. Or from the main branches. The inflorescence consists of male and female flower. The inflorescence consists of male and female flower, enclosed by a green leathery structure. Enclosed by a green leathery structure known as sheath. Green soft leathery structure, you can see. When the inflorescences are developed, then the sheath is splitted, release the male and female flower and droop off. Then after releasing this, it is droop off, then you will find two flowers. One is female flower, another is male flowers. Female flower is bigger in size, and male flower is small. Male flower surface is smooth, female flower surface is granular. At the anthesis and dehiscence, the male flowers, there is a development of one honey-like structures. Some honey-like structures develops, and the upper surface becomes sticky. Then there is a growth of the mold, good media for growth of fungus and mold. Growth of the mold and becomes black and rotten, then droop off. So, this is also one type, that is male flower, male inflorescence rot, but it is not a serious problem, because after dehiscence the male flowers droop off, then the female flowers remain in the plant in good intact condition. And these female flowers, bigger in size and having granular creamy white woolly structure, creamy white color and woolly structure, just like a wool's. And after food set, you will find the fruits fall, right? Then what are these spines? These are spines or rind. These are known as spine or rind, or in some books you will find wrinkle. So what is this? These spines. Here you will find hundreds of gynoecium. These all the woolly structures giving a granular appearance. These are only gynoecium stigma. And this is the style, and this is the peduncle, and ovary is there. So when there is a pollination and fertilization takes place, then there is a formation of the fruits. And there is a development of seeds. But other gynoecium which are not fertilized, they remain as such. That means when you will ripen jackfruits, when you cut it, dissect it, then the colored pulp you are extracting and use for table purpose. Seed is used for culinary purpose, cooking purpose. Then others, white structures, having this gum-like structures, but the white structure, these are all unfertilized style of the gynoecium. So if there is a good pollination and fertilizations, then the number of seed is more. If there is a lack of pollination and fertilization, the number of seeds is less. And this lack of pollination and fertilization is mostly depend upon the sunny conditions, nutrient conditions, and the healthy condition of the plant. Under heavy shade conditions, unfertile land, unmanaged conditions, soil conditions, then you will find few numbers of fruits with less number of seeds. So this is the, and this spine is the, what is spine? It is the stigma, stigmatic end that remain outside the skin, and at the maturity development stage it becomes black color. The crop is mainly used for table purpose. And also used for making squash, jam, fruit bar, candy, and wine. Nowadays, good wine is prepared from ripe jackfruit pulp. And this immature fruit and seed of ripe fruits are widely used for cooking purpose, culinary purposes. And the seed is a good source of carbohydrates. Okay, I will give you this composition. The composition of ripe grapes. That is carbohydrates 16 to 20%. TSS, total soluble solids, 25 to 29 degree Brix, written in percentage. Vitamin A, it is 500 to 580 international unit. Thiamine content, that is one of the vitamin B complex, that is 30 milligram per 100 gram of the pulp. Then pectin content 1.5 to 6%. Then iron content is high. Iron content is 500 milligram. Then phosphorus content is 30 to 40 milligram. 30 to 40 milligram. And calcium content is 20 to 30 milligram. So you see good amount of carbohydrates and vitamin A, highest vitamin A found in mango. Then next, ripe papaya. Then jackfruits. Good amount of iron is there.